There was a time when people thought that the characteristics of a species were fixed. That is, that species don't change over time, but that the world and all of its inhabitants were created just as we see them today. The idea that evolution results in the slow adaptation of a species to its particular environment, or the gradual accumulation of changes that increase the diversity of life on Earth, was not only laughable, but blasphemous. Today, only 150 or so years later, evolution is the foundation of the entire field of biology, and its offshoots of medicine, zoology, paleontology, and many others. We take it for granted that species change over time. Likewise, even up until the last few decades, the brain was thought of as a machine. Once its parts were fully developed in young adulthood, it was fixed for life. Of course, with a gradual decline in function as our bodies age. But today, we know otherwise. And just like evolution, the idea that the brain is plastic, meaning that structural and functional changes are in fact a fundamental property of the organ that gives rise to our minds, has caused a paradigm shift in the field of neuroscience. We call these changes neuroplasticity. We think of the brain as plastic or malleable rather than static and rigid, like plastic that can be molded into any possible shape. And this plasticity happens at many different levels, from the very tiniest changes on the brain cell walls to large, sweeping changes across multiple brain regions. As you can probably imagine, neuroplasticity captures our own experience of our minds much more accurately than the idea of a computer or other machine. Thoughts come and go, we learn and we forget, we improve and we regress. But of course, there are some functions that seem to be more amenable to change than others. It's easy to learn that someone has changed her phone number because she moved across the country. It's hard to remember what that new phone number is when we've dialed the old number thousands of times. And, as you're probably well aware, there are also times in our lives when our brains are more plastic than at other times. It's much easier to learn a new language if we've been exposed to it before the age of 5, when our brains are in what's called the critical period for language acquisition. But it's not impossible to learn or even master a language in adulthood. It just requires more effort. And admittedly, the process by which a new language is acquired in adulthood is a bit different compared with how it works in childhood. So what are the different ways in which our brains demonstrate plasticity? Let's start with the very smallest changes, which also happen to be the ones that have been studied the longest. These are changes that occur at the synapse, the gap between one brain cell or neuron and another. Brain cells exchange information by sending electrical or chemical messages across the synapse. A neuron is made up of 3 basic parts: the dendrites, the soma, and the axon. Dendrites extend out from the rest of the neuron like branches of a tree, and they receive information from thousands of upstream cells. This information from the upstream cells is sent down to the cell body or soma, where the nucleus, which holds the cell's genetic material, and other specialized parts of the cell, or organelles, carry out the metabolic and other functions to keep the neuron going. Finally, the axon. It's a single projection of the neuron, usually a long, slender nerve fiber, whose job is to pass along the information in the form of an electrical signal. Sometimes it transmits data a very short distance, as is the case when the neuron is in a tightly packed region of the brain. But axons can also reach very long distances, when the information needs to travel to and from the furthest reaches of the body, all the way up the spinal cord, or from the spinal cord deep into the brain. One example is the axon of the sciatic nerve, which extends from your big toe all the way to your lower back. The synapse is the junction between cells over which information is exchanged. An axon of one cell usually meets the dendrites of another at the synapse. But a synapse might also link a neuron with either an upstream sensory cell that has information about the outside world, or a downstream motor or other cell whose activity is controlled by the neuron. There are also synapses between dendrites of two neurons, or between two cell bodies, but these aren't as common. At the synapse, the electrical signal called the action potential from the upstream or presynaptic neuron triggers a cascade of chemical reactions that can have different effects on the downstream or postsynaptic cell. The electrical signal causes a voltage change that opens a series of gates called ion channels. These gates allow the passage of neurotransmitters, signaling molecules located inside the axon, to leave the axon and enter the synapse. In the synapse, the neurotransmitters float over to the other cell's wall and act like keys in a lock, binding to and opening gates in the wall. This initiates another series of electrical or chemical changes in that cell that affect the functioning of the downstream neuron. Some neurotransmitters excite the cell, triggering an electrical impulse, which we say causes the cell to fire. And some inhibit the cell or prevent the cell from firing or from sending an action potential down its own axon. The presynaptic cell turns its postsynaptic partner either on or off. And that, in a nutshell, is how much of the information that your mind is processing is propagated throughout your brain. But where does plasticity come in? Well, to answer that question, we must enter the fascinating world of memory, since forming memories is how we learn new things, of course, and one primary example of how synapses change. Let's start with the simpler types of memory, learning that two things are associated because they occur at the same time, or one predicts the other. Like how opening the fridge door will likely lead to the satisfying feeling of eating something delicious. Arguably, the most famous experiment demonstrating the power of associative memory is the story of Pavlov's dog, or dogs to be more precise. Ivan Petrovich Pavlov was a Russian physiologist who became a Nobel laureate at the turn of the 20th century for his work on the physiology of digestion. Despite this accomplishment, he's now most famous for his groundbreaking findings related to associative memory. In the course of his research on digestion, Pavlov noticed that his dogs would salivate at the sight or smell of food. The saliva helps the dog break down the food and digest it, so it's an important part of the digestive process. But he also noticed that the dogs sometimes salivated when there was no food present. Trying to decipher this enigma, he searched for the stimulus or trigger that caused that salivation. And he quickly realized that the dogs would salivate when they saw a lab coat. They had learned that experimenters wearing lab coats meant that food was coming. So then Pavlov, like any curious scientist, set aside his digestion work to sort out how this association is formed. He chose to create an association between food and the ringing of a bell so that he could control when and how the dogs were exposed to both stimuli. Pavlov found that when he rang the bell shortly before the food was served, the dogs would learn the association and begin to drool when the bell was rung, even if the food wasn't present yet. Pavlov called this new association a conditioned reflex, because it signals food, which elicits the automatic or unconditioned response of salivation. We now talk about Pavlovian or classical conditioning when referring to the association of a previously neutral object with a stimulus that triggers an automatic reflex, like the bell that becomes associated with food, which triggers salivation. And as any of you pet owners can attest, this type of reward-based conditioning remains the most effective way to train your dogs. Cats, well, we all know they are a special case. So how is this new association laid down in the brain? In the 60s and 70s, two neuroscientists named Turrier Lomo and Tim Bliss discovered that when two neurons that are connected by a synapse are stimulated at the same time, they become associated. This means that in the future, when one fires, the other is more likely to fire as well. We say that the synapse has been strengthened because the connection between these two neurons is now stronger. We also say cells that fire together wire together, which is shorthand for this type of synaptic plasticity. Let's say that there's a set of cells in your brain that turns on or fires when you see the refrigerator door. And there's another group of cells nearby that fires when you see any type of food. Now, whenever you open the fridge door, both sets of cells fire because you see the door and you see the food at the same time. Over time, these two cell groups fire together often enough that the connection between them gets stronger at the synapse. Just seeing the fridge door is enough to set both your food and fridge detector cells firing. The activity of these cells is now connected, and the fridge door is associated with food. This type of association, which can persist for weeks, months, and even years, was first discovered in the rabbit hippocampus. You'll remember this highly organized structure from our second lecture on the organization of life. The hippocampus is responsible for turning short-term memories, which last only a few seconds or minutes at most, into long-term memories that we can recall over the course of a lifetime even. Your ability to keep in mind a phone number until you dial it, or a set of directions long enough to make a few turns, are two examples of short-term memory. Long-term memories are memories for things like your wedding day, or your child's birth date, or how to ride a bicycle. Most long-term memories, especially those for facts like your child's birth date and events like your wedding day, involve the activity of the hippocampus. But how exactly do neurons wire together when they fire together? We call the facilitation of the connection between simultaneously active neurons long-term potentiation, or LTP for short. Because neurons become associated by making it more likely that if the upstream cell sends down an action potential, it will successfully activate the downstream cell, even over the course of weeks, months, and years. And it turns out that the mechanisms underlying synaptic plasticity are different in different parts of the brain. Even within the hippocampus, the molecular mechanisms underlying LTP can change. The neonatal and mature hippocampi use different proteins and enzymes to accomplish LTP. So now you see how this type of synaptic plasticity or strengthening might underlie how we learn, how we associate one thing with something else. The color red with fire trucks and stop signs, cinemas with the smell of popcorn, your partner with feelings of romantic love. Although we've been studying the molecular basis of new memory formation for decades, there are still many questions that remain before we have a full understanding of how memories are laid down in the brain. What does the firing of these neurons represent, for example? Different objects, different ideas, something else? How many neurons represent any given memory? Does the activity of groups of neurons underlie memory formation and retrieval? What happens when neurons die? Are the memories lost or transferred to other neurons? How does that happen? As is always the case in science, answering one question simply leads to a whole new set of questions, and neuroscientists studying memory will have jobs for many years to come. So we can observe plasticity at the level of the synapse, the tiny gap between nerve cells, but we can also see major changes with experience, even in adulthood, across large swaths of the brain. Often, you'll hear neuroscientists talk about maps, brain mapping, cortical maps, remapping, and so on. The analogy of a map is particularly appropriate when we talk about the brain, because we've learned that there is some topographical organization in terms of brain function. Different functions are localized in different brain regions. But the brain is also highly interconnected. We can't forget that no region acts alone. Some regions are more important than others with respect to certain functions. Like if you have a stroke in the front of your left hemisphere, you'll likely experience some difficulties with language. But those regions are also connected to your memory stores and your decision-making cells and various other parts of the brain. There are two regions of the brain, though, that have been very well charted and seem to be pretty consistent from one person to another and even between humans and other primates. These are the somatosensory cortex and the motor cortex, those parts of the brain that are responsible for your sense of touch and your ability to control the movements of your body parts. These two regions are located near the middle of the top of your head, and they take the form of two strips on either side of a very obvious landmark called the central sulcus. Sulcus means furrow or depression, and it looks like a large canyon that separates the front from the back of the brain. The motor cortex is closer to the front of your head than the somatosensory cortex, which lies behind the central sulcus. What's fascinating about these two strips is that the many parts and functions of the body to which they correspond can be found there lying right next to one another. And the more fine motor control or sensation that a body part requires, the more cortical real estate it occupies. The allocation of this real estate in the brain can be depicted in the form of a useful, if bizarre, human figure called a homunculus, whose various body parts are shown as larger or smaller depending on how much space in the brain is devoted to them. For example, our homunculus, which is Latin for little man, has very large hands. This is because our hands are very important to us, both in terms of functionality and sensitivity. So a paper cut on your finger is much more painful than one on your forearm. And we use our hands all the time for fine movements, typing, driving, dressing, cooking, and so on. So the hands are overrepresented in the motor cortex too, compared with, say, the torso. The torso of the homunculus is correspondingly small, and we can't feel things as well on our stomachs as in the palm of our hands. We also can't move our core as well as we can our hands or feet. Our homunculus also has a very big mouth, with the lips and tongue protruding almost grotesquely. He's not a handsome fellow. And of course, our genitalia are quite large on a somatosensory homunculus, but on a homunculus showing only motor functions, they're not very large at all. Our hands are even larger on a purely motor homunculus than on a sensory one, showing that manipulating hand movements is very important, even more important than feeling on the hands. And our motor homunculus, my friends, has an even bigger mouth, but skinny arms and legs and virtually no torso. But what happens when you work out or spend years training a particular skill? Does your homunculus change as a result? The answer is yes. The sensory and motor regions of the brain are plastic and change with use. How might this work? The answer lies in one of the most important discoveries in neuroscience, that if you prevent kittens from seeing the world during an early stage in their lives, they will be permanently blind. Research in this area started when American physiologist David Hubel and Swedish physiologist Torsten Wiesel studied the visual system in anesthetized cats. As I described in Lecture 7, they discovered in the late 1950s that the visual system is highly organized in the occipital lobe at the back of the brain. Remember the columns and the blobs and the feature detectors. One part of the cortex detects lines, another processes orientation, a third the movement of objects, and so on. Hubel and Wiesel also found that for certain functions, there is a critical period during development that, if missed, will result in a lifetime of disability. This principle was what won Hubel and Wiesel the Nobel Prize in 1981. In order to understand how the visual cortex develops, they took a group of very young kittens and sewed one eye shut on each of them. When they prevented the kittens from using one eye in this way, they found that the kittens never developed proper binocular vision, even when their closed eyes were later opened. Binocular vision is how we can see depth, because we can look at the different components of the visual field and see how the different eyes represent it. But what's really interesting about Hubel and Wiesel's discovery is that the brain region that would have supported vision in those kittens didn't just stop working. It was recruited to do something else. The brain rewired itself so that no cortical real estate was wasted. The columns of neurons that normally would be devoted to processing visual input from the deprived eye, as we learned in our lecture on seeing, were now processing information from the other eye. This rewiring happens at the synapse, with some connections between neurons strengthening and others being pruned or weakened. It also happens in terms of what part of the world the cell responds to. Its receptive field changes. It now seems intuitive that parts of our brain are wired to parts of our bodies and that these connections can change as we use our bodies in different ways. But the brain didn't have to work that way. In fact, for a long time before the plasticity of the brain was understood, neuroscientists thought that after childhood, each peripheral nerve outside the brain had a specific destination in the brain. Every point in your body that is connected to a peripheral nerve, enabling you to feel it, was thought to end up in a specific part of the brain. This makes a lot of sense. Just like information from your eyes always ends up in the back of your brain in the occipital lobe, so too should the peripheral nerves send their signals to a specific part of the somatosensory cortex. But in the late 1960s, Michael Merzenich, an American neuroscientist, along with a couple of neurosurgeons, wanted to see how peripheral nerves get reshuffled in the brain when the nerves are cut or injured. To that end, they cut the nerves in the fingers of a few monkeys and immediately sewed the severed bits together. They then remapped the somatosensory cortex in the monkeys, expecting to see a jumbled mess where the two fingers are represented, with some neurons getting signals from one nerve mixed in with neurons getting signals from the other nerve. But that's not what they found. Instead, once the monkeys recovered from surgery, they discovered a completely normal arrangement of brain cells devoted to the fingers. There was no crossing of wires, even though the peripheral nerves themselves had grown back crossed in the hands. Somehow, the brain had sorted out which signals came from which fingers. Michael Merzenich wrote up these findings in a paper and submitted it to its postdoctoral advisor. He realized he was suggesting a paradigm shift in neuroscience, that the brain was not static in adulthood, as was the mainstream view, but rather that it remained plastic, at least with respect to the somatosensory cortex. His advisor, Clinton Woolsey, however, vehemently disagreed and removed any reference to plasticity, stating that Merzenich had overstated his findings. But Merzenich was not finished with plasticity. He went on to start his own lab at the University of California in San Francisco in the 1970s and researched diseases of the ear. Under the guise of mainstream work on hearing, he was really interested in investigating whether the auditory cortex also showed plasticity, just like the somatosensory cortex. And so he worked with engineers and other scientists to invent a device that might restore some hearing in people who were deaf. Not just those who have partial hearing and can be helped with a hearing aid that simply ramps up the amplitude of sound coming into the ear, but people who have a damaged cochlea. The cochlea, you'll remember, is that part of your inner ear that translates sound waves into electrical signals that are then carried to the brain. Merzenich wanted to know if the auditory cortex could decode a device that replaced the cochlea and translated important sounds like speech directly into electrical signals. If so, then his success in restoring some hearing to profoundly deaf individuals would validate the idea that the auditory cortex is plastic, because the brain would be interpreting these new electrical signals from a device rather than from the cochlea. It's as though I replaced the hard drive in your computer with a completely different device, and your computer figured out how to make some sense of the signals. And so he did. The implant that he designed, along with other scientists and engineers, remains the inspiration behind one of the two leading cochlear implants to this day. But Michael Merzenich was not done just yet. To observe plasticity directly, he had to return to monkeys and the somatosensory cortex. And so, with colleague John Koss from Vanderbilt University, he conducted an experiment in which he completely severed one of the 3 peripheral nerves in the hand of a monkey. He wanted to know what would happen to the somatosensory map if one of the nerves was no longer sending signals to the brain. He was not surprised then to find that when he touched the part of the monkey's hand that had been previously represented by the severed nerve, the region of the somatosensory cortex that had previously mapped that area showed no activity. After all, no signals were being sent there. But when he touched other parts of the hand, he was flabbergasted. The part of the map that previously had been activated by the severed nerve was now firing in response to stimulation coming from nearby peripheral nerves. Those cortical maps for the nerves that had bled into the area that wasn't getting any signals. Cortical real estate had been reassigned. And so we come to the other side of neuroplasticity, what some neuroscientists call its dark side. It's the use it or lose it principle. If you don't stimulate a brain region or practice a skill for a long while, the brain will rewire to represent whatever it is that you are using it for. The brain survives on stimulation, and plasticity is a competitive sport. But wait, you might be thinking, people say that you never forget how to ride a bicycle, even if you don't ride one for 10, 20 years. Well, there's a difference between retaining a basic skill and a high level of proficiency in a complex one. The more complex the skill, the more subtle and specific the wiring in the brain that underlies that ability. You might be able to keep your balance on the bike, but you won't win the Tour de France again if you don't practice for a number of years, even if you're still in shape to compete. Unless you keep up your practice of bike racing or a foreign language or playing the violin, your ability will decline as the brain regions that changed as you develop the skill are co-opted for other purposes. The plasticity of our brains is also what makes bad habits so hard to break. The more often you do something, the more ingrained the wiring becomes for that particular activity. If you always use the wrong muscles to swing a tennis racket, the more you practice with the incorrect technique, the harder it will be to learn a proper swing. In fact, it's harder to unlearn bad habits than to learn new ones. This principle is well known to every coach and music teacher in the world. Once a student has developed a bad habit, it takes 10 times more work and time to fix it than it would have taken to learn the skill the right way from the beginning. But by the same token, with appropriate training and effort, we can mold our brains at any age to function more efficiently and more creatively, and we can develop skills that were previously thought impossible. We don't yet understand all of the mechanisms of plasticity, and as a result, we don't know what the limits are of our ability to change our own brains. Perhaps our capacity to improve is, in fact, limitless. In the next lecture, we're going to delve more deeply into the implications of neuroplasticity in our lives, and we'll get to the question of how to harness neuroplasticity to stave off the cognitive effects of aging, to recover from disease, and to master complex skills.